And now we go on with one more important plant in Malaysia, where we call this as waste product, paddy waste product, where the rice is our number one food in Malaysia. We always take rice every day, but we throw the waste product, what we call as the husk and also the straw and also the husk of the rice, where we will throw it away. In Malaysia, we throw the waste product is about 7 million tons per year. In India, it's about more than 120 million tons per year being thrown. So what we have from this waste product, we found vitamin E, we found tocotrienol, tocopherol that exists in the plant, this waste product. So we start to study with our in vitro data through one of my PhD student, Dr. Morgana, and he found that there is a anti-preventive activity with the paddy husk. As you can see on the slide, we get the IC50 and how the cell change, the phenotype of cell, the one exposed to the paddy husk being killed. There is apoptotic antibody activity being found. Also with the straw, also we found the IC50, the paddy straw, also showing the anti-cancer activity and still give the good of promising cell death morphology. Here we use a scan electron microscope on on prostate cancer cells. This is DU145, which are androgen dependent cells. You see very clearly how it look like, and when we expose to the when we treat it with the paddy husk, it become shrinkage. You see, it shrinks clearly, so means that the paddy husk have anti-proliferative effect. Okay, here also the same thing. Okay, all the membranes being ruptured. And when we do our Western blot analysis on straw and husk, so what we found that it induced the caspase 9 and also reduced the BCL2 and also BAX proteins. Same go to caspase 3 and also BAT. So here we draw, okay, we try to draw the pathway that might be effect to DU145 cells of prostate cancer, maybe through caspase 9 to induce the apoptosis. This is our prediction. So here again we do safety study, where we want to see whether there is what you call toxicity or not. What we found, there's no toxicity condition with the fibroblast cells. Same go to the straw. And we study in animal model in vivo as well. And here this is a body weight result. There's no change of body weight. We compare the husk and also with the asper we give to the rats. Okay, the asper extract as well as the paddy waste product. And you see the consumption of water, there's not much change, and the food also there's not much change. And the blood pressure, these rats have high blood pressure, about 200, more than 200 blood pressure milligram. And you see the blue color here, is the high blood pressure with the SHR rat, spontaneous hypertensive rat. And you see the straw and also the husk give about 140 at the end after 16 weeks of drink the paddy waste product. And with the asper, it go more or less about 120, 130 of blood pressure. When we study blood pressure with the prostate, because there is a clear evidence the relationship of hypertension and also the benign prostate hyperplasia, where in clinically doctor will prescribe alpha blockers, terazosin, prazosin, which the one can reduce the blood pressure. It also can reduce the benign prostate hyperplasia. So therefore, in this model, the rat also giving high blood pressure with the benign prostate hyperplasia. Therefore, we study the extract on this particular model, whether it works or not. So you see the blood pressure going down. This is the relative body weight. There's not much different in the body weights or the tissue weights. There's a little bit of reduction. But the most important here is that the SC9, okay, the epithelium area of the SC9. If you see the benign prostate hyperplasia, the SC9, the epithelium area is more bigger, it's much more higher compared to the normal one. It's very thin of epithelium area. But the one we expose with the ESPER as well as the Paddy waste, it show like a normal WKY result, the normal hypertensive rat with a normal prostate cells. Okay, you can see here, this very clearly show very good results.